Hello and welcome to Aurelian University. In this video we're going to talk briefly about the difference between gram-positive and gram-negative bacteria. So really quickly, essentially what the difference between gram-positive and gram-negative bacteria is the size of this layer of peptidoglycan and where it is relative to our membranes. So in gram-positive bacteria we have our simple membrane with a phospholipid bilayer, and then we have this, in green we have a layer of peptidoglycan, and it's going to have lipoteichoic acid going through it. Now just like any normal membrane, you can have integral proteins inside the membrane itself, but lipoteichoic acid is going to be primarily in the peptidoglycan layer, and it might extend a little bit into the membrane, but it's always going to be at least partially up here. In a gram-negative bacteria, we're going to have this phospholipid bilayer. Then we'll have a much smaller peptidoglycan layer that's going to be a lot thinner, and then we'll have a second membrane actually, and then around that second membrane we're going to have these, which are lipopolysaccharides, and those lipopolysaccharides are going to attach to this second membrane, but they're also going to sort of protrude out. So that's the main difference between the two. And what this means is that when we use the gram stain, we'll see a slight difference in color. Well, not really a slight difference, a very big difference in color between the gram positives and the gram negatives, because the cell walls, since they're structured differently, will cause some of the bacteria to retain different dyes than other of the bacteria. So we'll have these purple ones, and then we'll have these sort of pinkish reddish ones, and these are supposed to be purple. I didn't have actually a purple pen, so they're actually just blue Sharpie and red Sharpie. But over here we have our red Sharpie ones, and this is why this is where the terminology gram positive or gram negative comes from, because the gram staining technique is used to differentiate between them when looking at them under a microscope.